Okay, welcome everyone. My name is Jessica Liu, and together with Christina Schultz, we're happy to launch the first webinar of the Benthic Ecosystem and Carbon Synthesis, or BECS, working group activity. And to start us off, I will ask everyone to participate in a Mentimeter poll. So the way this will work is go to this site, www.menti.com, and put in this code 42851861 when prompted. And there will be two questions, but we'll first start with this one. May will also put in the link on the chat. Just to get a sense of who is here, what your expertise is, and then we'll ask a little bit about, what you work on. Great, this is looking like we have a pretty diverse crowd here. And it looks like the responses have slowed, so I'm going to move to the second question. So your screen should have refreshed or changed. And then here we're asking for just short answers on, what part of the world you work in and what tools you typically use. This is great. There's lots of diverse answers. This is really fun to even just look at.
I think this is the most diverse answers I've seen.
Yeah. Lots of people working in Europe. Some folks global versus regional, eddy covariance. Lots of Chesapeake Bay, sediment cores.
I think these are just all the answers from Dave Burdage.
All right, looks like the answers have slowed. So going back to our slides. So I'd like to introduce the organizing committee of this working group. Christina Schultz is the lead. She is an assistant professor at Northeastern University who works on biogeochemical cycles and regional global modeling. I am a research oceanographer at the NOAA Geophysical Fluid Dynamics Laboratory. And I work on marine ecosystems and Earth system models. The rest of organizers, if you're here, can I ask you to turn on your videos? Wally Fulweiler is at Boston University and works on coastal biogeochemistry and nutrient fluxes. Matt Long at Woods Hole Oceanographic Institution works on benthic oxygen and sensor development. Colleen Petrick is at the Scripps Institute of Oceanography and works on fish models and benthic fisheries. Jeremy Testa at University of Maryland works on benthic and coastal models. And Damian Brady from University of Maine works on benthic models and ocean carbon dioxide removal. All right, Christina.
Yeah, I'm just going to ask you to change the slides.
Okay.
So I'm just going to give a quick introduction of what are we doing and where we're coming from with this and some of the other OCB synergistic activities. So for those of you who are not familiar with the OCB, it's Ocean Carbon and Biogeochemistry program. And what OCB is focused on is understanding the different parts of the carbon cycle in the ocean and the influence of climate and human-driven changes on this carbon cycle and of the ecosystem, how this ecosystem influenced the carbon cycle. So Jessica, next slide. So we're building on some previous OCB activities that have been done in the past, particularly CCARs, or the coastal carbon synthesis that ran from 2008 to 2016, and was an activity partner between OCB, the North America Carbon Program, and the US Carbon Cycles Science Program. And what they did was divide different working groups that were focused on different regions, so East and West Coast, Gulf of Mexico, Arctic, and Great Lakes. And they had a bunch of meetings to try to synthesize data and model the different components of the land to ocean aquatic continuum. So this figure is just showing a schematic of the fluxes that CCARS was focused on. So how does carbon move from rivers to the open ocean, and what kind of transformations happen in this and along the way. And just to give an example, for the East Coast, in the 2012 paper found that the benthic fluxes were among the least constrained fluxes. And this is a figure from a synthesis put together by Ray Najar in 2018. That's an update on the 2012, and there are values for the benthic fluxes, but what they found is in the East Coast is that they were still below the global average, the fluxes between the water and the sediments, and that these low fluxes might actually reflect a spatial variability that needs to be accounted for. So there was a variety of values within the observations that average to that, but we still need to understand the spatial and temporal variability of the benthic fluxes. So next slide. Among some of the recommendations from CCARS are accurate measurement of sedimentary processes and fluxes of carbon-relevant species across the sediment-water interface, and the use of biogeochemical models to scale up from the limited observations we have. So next slide. So some of the other synergistic activities that OCB has going on recently are the seesaw. So seesaw is focused on extreme events and how different extreme events, so fires, hurricanes, are going to affect the carbon cycle in the land to ocean aquatic continuum. The ocean carbon dioxide removal. So if we want to remove carbon from the atmosphere and store it in the ocean, I think it's important that we understand how carbon is going to be processed in the benthos. And the fisheries and carbon working group that also is interested in trawling, export of organic carbon, and benthic ecosystems. So next slide. So the BECS goals, what we're trying to do is facilitate communication and data sharing between benthic, coastal, and global biogeochemical modelers and observationalists, establish some shared data protocols, including some common definitions and variables, names, and units, and develop products that could be used for model development and validation, as well as some recommendations for future directions in observation and modeling. So this is our first webinar. Throughout the next 3 years, we're going to do a series of webinars focusing on different aspects of this benthic pelagic interactions. And we're also going to have 2 different workshops to define research priorities, do the data synthesis, and identify methods and future directions for benthic research. So this was just a quick introduction to tell you where we're coming from, and it's my great pleasure to introduce Dr. Jack Middleburg. Dr. Middleburg has done extensive work on benthic processes and benthic pelagic interactions at several different spatial and temporal scales, and he has won a series of awards. More recently, he's been elected a fellow member of AGU. and a member of the European Academy of Science. So he's going to give us an overview of all the benthic research. With that, Jack, please take it away.
Oh, you're muted, Jack, if you can unmute.
Now I should be, now you can hear me.
Perfect.
Yes? Okay, sorry for this. First I would like to thank the organizers, in particular Jessica and Christina, for asking me to give this presentation. And then, okay, I'm invited. What shall I tell in 20 minutes to an audience as wide as we have seen with Mentical, that basically we have interest in many things. So that's also my frustration is my scientific life, that I'm interested in too many things. So I've decided to focus on benthic pelagic coupling and on carbon cycling, and not discuss oxygen, nitrogen, and all the other things. So what I would like to, why am I not able to go further? I'm not, sorry, I cannot.
Try clicking in the middle of the slide with your mouse first.
Okay.
and
Okay.
then-
Okay, yeah, thank you. So I would like to start very wide. I together with some colleagues, I have been working on a carbon cycle for the fresh water, and we have an integrated picture of the terrestrial marine carbon budget, how it was about a century ago, around 1900, and how it was around the year 2000. So in around 1900, we already as human perturbed the carbon cycle with 1.85 petagram per year. Part of it already was accumulating in the atmosphere. The CO2 was already higher than the pre-industrial. We saw that there was an atmospheric transfer to the land of about 3.7, and only about 0.37 really accumulating on land. The majority was transferred to the freshwater, it's about 3.3. Then about 2/3 was being emitted again as CO2 back to the atmosphere, so an internal cycle. There was some accumulation in the freshwater. And then we had a transfer, which is most important to us, oceanographers, of about 0.87. And at that time, at least according to our model world, is that the ocean was a source, because basically 0.87 came from land and 0.5 accumulated, so 0.35 went to the atmosphere. So what we saw when we ran our model, which is basically an integrated assessment model, a water routing model, and then a biogeochemistry model for the terrestrial domain, and then we combine it with the Friedlichstein numbers for the global carbon cycle. We see that in the year 2000, we have a much larger human emission. We get accumulation in the atmosphere, as we all know, from the well-documented CO2 increase. We see that there is a larger flush from the atmosphere to land, and that there is more accumulation on land. There's also somewhat more accumulation, so transfer from the land biosphere and the humans directly to the freshwater, but that's only a little perturbation. And that's then the majority of it is then emitted as CO2 back to the atmosphere. A little bit accumulates more in freshwater because we have constructed reservoirs and all those things, but that's all very limited. And if you look at what we think is the best estimate for the perturbation of the river transport to the ocean, we see that very little changed. That basically, first, that the freshwater carbon cycle was not so much perturbed as we initially thought, say 15 years ago, and that the land-ocean transfer is quite robust. And of course, the ocean changed function from a source to a sink of CO2, as we all know. So the take-home message is here that inland waters receive more carbon. More of it was buried, and in particular more was emitted, but the export to the coast remained rather similar. The problem with a lot of the studies on the freshwater, the inland carbon cycles, that people don't distinguish between natural and perturbed carbon cycle. And as we all know that the background carbon cycle says that the ocean is a source of CO2 to the atmosphere. What I'm now going to take you on is that rivers deliver organic and inorganic in dissolved and particulate forms and nutrients to the ocean, and that of course fuels a lot of the biological cycles there. So let's first look at the role of the seafloor in terms of inorganic carbon. If you have been lecturing and using the textbook Burner and Burner, you know already for 20 years that we have an issue here, that on the one hand, our conceptual picture is that the alkalinity delivered by the rivers should be balanced by calcium carbonate burial in the ocean. And if you make this budget, and the numbers, some numbers are quite robust, some others are a little bit more debatable, but overall you see that about twice amount of alkalinity is buried relative to what's coming in. So we really have an issue here, and this is already known for quite some time, but we tend to ignore it, in particular the modeling community and the oceanographic community. Of course, given the residence time of alkalinity in the ocean, you would expect that there should be some balance. And one thing in a review I wrote a couple of years ago, we identified that there should be also inclusion in the budget of the particulate inorganic carbon, because if you remove particulate organic carbon, you should also include the sources of particulate inorganic carbon. The initial estimate was a little bit higher, but one of our colleagues in Utrecht, Gerrit Müller, he made a better estimate that we now think it's about 10 units of alkalinity are being delivered, so that we now have an input of which is higher, but still not enough. And we also included all the other potential sources of alkalinity, like submarine weathering, anaerobic process, organic matter burial, and we are getting closer to balance given all the uncertainty, but we are not yet there. So I would like now to continue a little bit in the direction of the calcium carbonate cycle before I go to the organic carbon cycle, and talking about carbonate dissolution and the role of the ocean floor, because that's what we think is the common denominator among most of us here. So this is work done by Oliver Sulpus, who was in Utrecht till a few weeks ago. He's now in Serres in France. He requests excess alkalinity versus the age of seawater, and estimated in that way the dissolution of calcium carbonate in the water column, and combined with estimates of seafloor dissolution and seafloor burial. And in that way, he back calculated that there should be about 76 TMol of calcium carbonate being exported, of which about 30% is buried, about 47% dissolves in the water column, and 40% dissolves at the seafloor. So in terms of calcium carbonate, the seafloor is really important. This calcium carbonate was not specified what it is, whether it is magnesium calcite, Aragonite, or calcite. And we all know that the saturation levels, which you see with yellow, pinkish, and blue colors, they of course are different. And that's something which we should do a better job in the future, because if you look at what's the effect of this diversity in calcium carbonate minerals on the benthic ecosystem, we did some computational work where we added a pteropod on the seafloor, and we did grain scale calculations, and we saw that if you add just one pteropod on all those calcite grains, you see that the dissolution of calcite at the sea bottom really declines a lot if there is an aragonite particle there. Now, given that aragonite fluxes are similar to calcite fluxes, at least in the surface, that there is molecular evidence for arrival of pteropod material at the seafloor, molecular biology evidence, I mean, and also that there is evidence that there is Aragonite in deep sea sediment traps. We think that Aragonite reaches the seafloor at many places, but we don't see it because it dissolves, and as it dissolves, it preserves a little bit the calcite. And you can see that if you we made a tentative we made a suggestion on how important this process can be. And if you look at this picture here of the global map, where you see in blue, you see basically the seafloor, which is in between the calcite saturation depth and the carbonate compensation depth. So that's basically within the calcite lysocline. And you see that if you do the zonal averaging, you see distinct maxima, and those correspond with model estimates of the carbonate fluxes in terms of calcite and in particular aragonite. So it may be that we have to make our models and on this of carbonates cycling in the ocean a little bit more complex than just looking at one carbonate phase, because it can be that a small flux of aragonite material reaching the deep sea floor impacts the preservation of calcite. So the take-home messages for the time being for the calcium carbonate is we have an alkalinity balance. If you want to have that, we have to include river and particulate inorganic carbon input into our budget, and also add additional alkalinity sources, and those are mainly covered by the organic carbon cycle, how much is happening anaerobically, how much is preserved. If you look at the carbonate export, we see that only about 13% is buried and 40% dissolves at the seafloor. And we see that there's surprisingly little information about aragonite versus calcite export and fluxes. And yet this may be very important if we want to advance our understanding of calcium carbonate dissolution at the seafloor. So this was the calcium carbonate. I returned to it in the modeling section. Now a little bit about oxygen consumption at the seafloor. So on the left, you have the conventional traditional knowledge. We have about 50 petagrams of primary production. We get an export of about 10, and then only about 2 units are reaching the seafloor, and only about 0.2 is being buried. Those numbers, of course, are not that nicely rounded off, but I always use this 50, 10, 2.2 rule when I'm lecturing, because those numbers are more or less right, and they're easy to remember. So that's one part of the global story. The other thing is, then we go to another textbook of Emerson and Hedges, the chemical oceanography book. They have a very nice figure about the respiration processes, whether that's happening in the water column or whether that's happening in the sediments. And they combined observations, if I recall, from Rick Janke for the sediment with the water column respiration, and then they saw that somewhere at 3 kilometers you get that the benthic respiration becomes more important than the water column respiration, and the main reason is, of course, the hypsometry of the ocean floor. So the overall picture is that in the upper 3 kilometers of the ocean, the most of the respiration is pelagic, but there is, of course, benthic respiration, and at depths more than 3 kilometers, basically, benthic respiration can explain most of the oxygen consumption. So I wanted to see whether we can do this with a consistent set of data, because Emerson and Hedges use 2 different types of data. So I used observation of sediment oxygen uptake at the seafloor. I used the data from Ronnie Klute and others, and I basically said, okay, if I make a very simple model of the ocean where you have 4,000 layers like this, and you include the hypsometry of the ocean, and then at a certain moment you get a flux, it reaches a sediment, and then it is being respired, or it goes into the water column, and then it is respired in the water column, etc. So you can work this out, and then you can calculate the respiration in the ocean. So this is water depth, this is respiration in kilograms per year. And what I've done here is based on these type of calculations, I calculated the amount of respiration in the water column, entirely calculated from sediment oxygen uptake data, which is this dashed line, blue dashed line. And then I compared it with the respiration, which you can estimate by taking the divergence of either the Martin curve, say the sediment trap data, or the Suez curve, the sediment trap data. So really the two first publications reporting the attenuation of the flux in the ocean. And you see that we get a very similar trend, just using only benthic observation. But we do see that the benthic observations imply a somewhat higher respiration rate. And for those of you who have worked on consumers in the benthic domain, we often see that in deeper waters, we as benthic observationalists, we always get too little carbon from above. And this is one indication that you see if you do the calculations from the sediment side, that there should be more respiration in the ocean than we thought before based on the water column. What we also can see is on the figure on the right here, we see that this figure, we see that if I plot then the respiration which is happening in the sediment, the respiration which is happening in the water column, and the total respiration, you see that above 3 kilometers it is dominated by respiration in the water column. Below 3 kilometers it is basically run by sediment respiration, and this is fully consistent with, say, the textbook picture of Emerson and Hedges, but calculated in a systematic, independent way. So this is for the global ocean, and one of the problems with this approach is that in this simple box model assumes basically that the whole ocean is laterally homogeneous. And of course, the sediments at a depth of 100 meters or 500 meters, you don't find in the center of the ocean, just at the margin of the ocean, where usually the primary production is somewhat higher. So you can have a bias. So for that reason, I showed this picture, which is work which Oliver Sulpers did with me, where we estimated water column respiration, that's the blue, by basically following oxygen concentrations, just the measured oxygen concentration, or the apparent oxygen utilization, or other properties of oxygen. Doesn't really matter so much. Versus the water age, the tracer age of the water, and then we can estimate the amount of respiration, and we do it for 6 parts of the ocean, the subpolar, North Pacific, etc. You can read them here. And we also have the sediment oxygen utilization rate, which is based on the data of Biogeochemistry of the 22 review article, which takes into account local primary production, so it gives some regional differences. But overall, you see that we basically get the same picture again, that at the water depths more than 2-3 kilometers, all the oxygen depletion in the deep ocean you can explain by just benthic consumption. So the take-home messages from this observational site is that carbonate dissolution 40% happened at the seafloor, and it mainly occurs at larger water depths. Organic matter mineralization about 20% at the seafloor. The intensities of mineralization are the highest at shallow water depths, as we all know, because that's where most of the carbon arrives. But it also shows that oxygen demand in the ocean at more than 3 kilometers can be fully explained by what's happening in the sediment. So that's what you can get out of observations. Now we're going to look at how can we make models for this. And this is something I did together with my colleague Kalin Sutat in the mid-90s and 2000s, where we developed coupled benthic pelagic models for various systems. And one of the things which we first figured out is what are the models there, what are the approaches, and basically we recognize 4 levels of complexities, and I will go to them in a minute. So the benthic pelagic coupling is not straightforward the modeling. That's why you see so much variability, so many different approaches, because if you look at the sediment water interface, you get a few things there. You get a timescale difference. Things happening in hours, days to weeks in the water column. You go into decades to millennia in the sediment. If you're looking at particles, particles are settling, so an advection dominated in the water column. Then they come in the sediments where they are biogeochemistry dominated. And then if you come below the bioturbated zone, you again are in an advection dominated system. The other thing is that you are going from usually an aerobic water column to aerobic, anaerobic processes in the sediments, which have consequences for many things about what's happening with the nutrients, whether you produce alkalinity or not. And of course, the sediment water interface and particularly the bioturbated layer determines what's the eventual transfer to the geosphere. So this is computational quite challenging because the turnover of hydrogen or protons like pH, that's happening in minutes in sediments sometimes. And some other things are turning over in thousands of years. And then you have to connect that with the water column. So we introduce 4 levels where basically the 4th level is the most advanced, where you basically have a fully developed physical biological model in the water column and a full diagenetic model coupled to it. But this is computational, very demanding, and not many people do this. Then there's a level 3 where you basically say, okay, I'm going to lump a lot of the sedimentary processes. I'm going to follow more or less the stocks of the particulate matter, and then I'm going just to model the pore waters. And then this is the method which is mostly used nowadays by coupled models, where you have a reflective boundary. Basically, the organic matter reaching the seafloor is immediately transferred into dissolved component. And this is the model which unfortunately was used a lot in the past, where you fix your concentration or fix your flux and you don't have mass conservation. You can see that here. We have the levels, the level 4, 3, 2, 1. The mass conservation is happening with levels 2 to 4. If you're looking at the delay, so the dynamics, you have to go to level 3 and 4. If you look at transformation about, for instance, how organic matter is transformed in CO2 and nitrate, ammonium, etc., or carbonates into alkalinity, you can do at level 2 and 3 to a certain extent. And then there are some issues like what you can do and what you cannot do. But the take-home message here is be careful with level 1. If you fix your concentration or your solute flux, you're losing or creating artificial mass, which has consequences for long-term model runs, and you have to restore, and you get some fluxes which are not really existing. Okay, some examples of those approaches which have appeared in the literature. So one of the first approaches, at least which we were involved, is that we ran a fully diagenetic model for carbon and nitrogen cycling in the deep sea. And then we derived a statistical model, which you will now call emulator. In the past, you would call a meta model or statistical model. You can do the machine learning, where we saw that if you have the full diagenetic model and you make a much simpler model, you see that you can get very nice prediction at a global scale. And more recently, it has been done by Belgian colleagues on a regional scale, where you have the nitrification and denitrification here by the full diagenetic model or by the statistical model. You see that you almost get perfect agreement. And then you can apply it in a 3D ocean biogeochemistry model, like here was being done for the Black Sea. So this level 3 is quite okay doing those type of things. And the advantage, of course, is that it's computational, cheap, and quite easy implemented. There are some mixed level 3s which people have explored, like, again, Belgian colleagues, they, rather than using a full numerical sediment model, they used an 2-layer, and also they have done 3-layer analytical models where basically you used analytical solutions to the diagenetic equations. And then you can do fast calculation and you get very similar results to the full numerical model. And then on the right, you see that something I did with Kalin Sutat about 20 years ago, the calculations where we have, on the right, we have a level 4, where we have a full one-dimensional physical biological model where we have a turbulence closure, we have aggregation of particles, unbalanced growth of the algae, etc. And then we have it coupled with a full diagenetic model. So we were able to do this. At the same time, we also realized if you're going to the shallow environments that our vision of the benthic system as just receiving things from above and exposed to bottom waters, that's too simplistic. So we also made some kind of level 3 model to include benthic primary producers, to include sandy permeable sediments where the current velocity or wave activity causes flow of water to the sediments, which has consequences for the mineralization. And we also included filter feeders, which were capturing particles from the water column. And we then looked at the benthic pelagic coupling. The take-home message is that there was not so much difference for the pelagic dynamics, to be honest. This was all about organic carbon. Bernie Boudreau has been promoting to do a similar thing for the calcium carbonate at the seafloor. Calcium carbonate, for the more chemists among you, of course, we all know that it is mineral interface controlled dissolution. But if you're looking in nature, you see that you see that in marine sediments that the dissolution is often controlled by the mass transfer through the diffusive boundary layer. So most of the seafloor, but also a small part like this, is controlled by mass transfer rather than chemical kinetics. And this then offers the opportunity to make simple models for calcium carbonate dissolution at the seafloor. Oliver Sulpus, when he did his PhD with Alfonso Mucci and Bernie Boudreau, made a model which was published in Proceedings National Academy of Sciences. And but they basically took the undersaturation of the bottom waters, combined it with the mass transfer, which is based on the current velocity at the seafloor, and they could model dissolution in the ocean. So this brings me to the big chance if you want to couple benthic and pelagic models. So the benthic models, I would say the diagenetic models, they really were developed by Bob Burner and his all the people working with him, analytical, and then we had the numerical models, a series of models in the 90s, and those models reached the scale of sophistication that they are good enough to be predictive, but they are one-dimensional. They're operating at centimeter scale, and they either they have seasonal time scale or steady state. If you want to apply them globally, you just get information at one spot at the seafloor, and you have to go global, and then you have to see how you can do this. But luckily, if you really play with those models, you see that usually only 5 or 6 parameters really matter. Some of the models have 20, some models have 50 to 100, and the majority, usually more than 80%, but for some of them 95% of the parameters, you can take uniform, globally uniform values. You don't have to fine-tune, so you can apply them and you get reasonable estimates what's happening at the seafloor. But there are some parameters which are really critical, which should be site-specific. Either they should be feed in from the water column, or you should use empirical relations or maps. Now, the first thing is the quantity of deposited material, either the flux of organic carbon, of biogenic silicon, or calcium carbonate, depending on where you're interested on it, which makes sense. If you are working with organic carbon, And also you have to look at what's the quality of the organic carbon flux coming in. And if you're working with carbonate, you have to look at whether it is Aragonite or Calcite. The next type of information you need is the bottom water concentration of oxygen, nutrients, and your CO2 system. So that's of course something which is also available from the water column models. And then the third one, which is a little bit more complex to obtain, is you need, if you're working with organics, you need biotubation and bioirrigation, because biotubation is the mixing of the sediments by the animals. It's the most important transport process at the seafloor. And mass transfer is needed if you basically the transfer of your dissolved substance to the diffuser boundary layer is limiting. You also need that information. So if you know this is a little bit provocative. I admit this. I want to make it very simple so that we go forward rather than losing ourselves in detail about an inhibition factor for sulfate on denitrification, which is one of those parameters which you can take universally constant. We can say that if you want to make a simple model and you're making a model for organic matter mineralization, you should look at how much carbon arrives at the seafloor, what's the quality of the organic carbon, what are the bottom water concentration of oxygen and nitrate, and you have to know some knowledge about biotubation and bioirrigation. And all the other parameters, they are secondary, tertiary importance. For the calcium carbonate dynamics, the same thing, the flux of carbonate to the seafloor, so basically how much carbonate is there at the seafloor, whether the bottom waters are undersaturated or not, and the mass transfer coefficient are important. So that has to do with basically the substrate, the chemical environment, and the physical transport. Those are the three most important things which should be in any model for benthic pelagic coupling. Now, my last slide is if you're going into shallow waters, because so far I have been focusing mainly at the global scale. If you're going to shallow waters, we have a few additional challenges. So basically, if you're going to look at the deep sea, we have a system which is donor controlled, as ecologists would say, is that material is settling and the deep sea communities cannot really do anything about it. They just have to accept that the material is arriving or not. That's basically how most of our benthic pelagic coupling is conceptualized. If you are in the more shallow waters, and about basically 1/3 of our continental shelf light hits the bottom and supports primary production, you have another situation is that the consumers, so the grazing herbivores in the sediments, they impact the organic matter producing machineries, the microphyta benthos, and this has a different dynamics. Then that's this way, and then the last thing which is also important in shallow sediments without large organisms standing out, which is number 4 and 5, which I will discuss in a minute, is if you have a system where there is a lot of wave action occurrence and that the upper part is continuously resuspended, deposited, so that you get lateral transfer of organic carbon, which happens a lot on the continental shelf. Then if you have biotubating organisms which are continuously feeding here and mixing material downwards, you get that there is a drawdown of material from the surface layer into the sediments, and this is really something you should incorporate it if you want to study benthic pelagic coupling. And then there are along the ocean margin and in a shallow system, you have a lot of suspension feeders, which can be the bivalves, it can be coral reefs, and those really live on the flow, the lateral flow of particulate organic carbon, which they then capture, and then via their biodeposition or feces production, they transport a lot of organic matter to the sediments. And then the final one is that there are some groups of organisms, particularly the sponges, that have been shown to be able to consume dissolved organic carbon transfer it into particulate organic carbon, which is then available for the rest of the food web. So these, say, mechanism 2, 3, 4, 5 are usually not incorporated into benthic pelagic coupling. There are some studies which do it. They have the additional challenge that, in particular for 4 and 5, is that you cannot assume steady state for the actors. You have to the consumption by the benthic consumers, they also lead to the growth of those organisms, which you also have to incorporate, and they also impact the physics by positive feedbacks. That if you get a coral reef, a cold water coral reef, or a tropical coral reef, they impact the physics, and in that way you get additional carbon flow. So that's a lot of challenges ahead. But with this, I would like to stop my presentation, and I'm happy for questions.
Thank you so much. We can take questions now. I see that there's some questions in the chat. Lisa Levin, would you like to unmute yourself and ask?
Sure. Hi, Jack. Thank you for a really interesting talk. Nice to see you. I wanted to ask, what do you think are the major seafloor observational gaps that need to be filled to enable modeling at the global scale? And I'm asking on behalf of the deep ocean observing strategy, which is really trying to identify what the observational needs, major observational needs are going forward in deep water.
Yeah, yes, thank you, Lisa. So one of the big issue, which is already around for quite a long time, and yesterday I was at a PhD defense in Utrecht about cold water calls. If you look at a lot of the benthic budgets which are being generated, and if you look at the carbon flux going down, which are being measured with sediment traps, there usually is not enough being supplied to the seafloor. So, and this is a long-standing question, and we do not know how to solve it very well. And of course, there are uncertainties. We know the under and over trapping of sediment traps. We know that there are lateral processes, but this is a big uncertainty, because if we do not know how much carbon arrives at the seafloor, we have an issue calculating about how much nutrients are returned, etc., because the amount of carbon arriving at the seafloor basically is the single most important factor in determining what the seafloor will do in terms of what lives it supports, what is being returned. So that's one thing which we should really do there. Then, of course, there are many related questions about how valid is say the assumption of steady state. Should we really resolve seasonality, the pulses, the spring bloom pulses? If we are going to look more about long-term impacts, the studies what people have done, making disturbance at the seafloor, how long does it take to recover again, which is very important if you're going to use or to preserve our seafloor. And that's also to do whether is the response time, is it 1 year, 10 years, or 100 years before the system is back to a steady state. And that's something which is another important thing, which is difficult to investigate, I know, but those are important questions.
I'm just curious, do you think dark carbon fixation is part of that gap?
No, I don't think so. If you dark carbon fixation is basically the energy is coming still from elsewhere. So if you sometimes people are saying, yes, we have dark carbon fixation, which brings additional carbon, but it doesn't bring additional carbon unless the energy is coming from elsewhere. If it is coming from the geosphere, so methane seeps, etc., yes, then it brings additional energy. But if most of the dark carbon fixation in the ocean is because there is organic matter degradation, there is reduced metabolites, and those are then oxidized and leading to carbon fixation, but that's not generating new energy. That's basically, it's not solving the problem. It is producing juicy material in the deep sea, It's producing specific types of organism, which specific organism might go for. So whether you have more archaeophory versus bacteriophory, those type of questions, that is important, but whether, I don't think it solves the carbon balance there.
Thank you.
Any other questions for Jack? We have time for discussion. This is part of the plan. Okay, I'll go ahead and ask a question while people are,
Nice.
so aside from the sponges and the corals and, how much ecosystem complexity do you think is needed?
That's a very good question. It is in the. So if you look at the maps of now we have more submarine observations, we see that there is more and more sponge ground, cold water corals, and all those type of things. And yeah, whether we should resolve them depends on your research question. If you're interested in those ecosystems, yes, we should do something about it. We should figure out how they can be sustained in the deep sea. Whether they are important for the global biochemical cycles, it really depends on their metabolism, which we have very limited information at a larger scale. There are now some eddy correlation observations. There are some upscaling, but then you have a lot of times, a lot of multiplication before you get a larger number. And usually the activities per square meter are about double in those systems compared to the background. So they do matter. I have made once a back of the envelope calculation for cold water corals that they do matter, but there's some work to be done there. Whether we should include them. If you're going to make a model for a real coastal system where there's a lot of mussels or oysters, yes, you should include them. So the work with Moni Smith and colleagues did at Stanford, that's really important. If you're going to some of those tidal inlets where basically those bivalves govern the carbon and nitrogen cycle, that's really important. I see there's a question in the chat from Ellen Kensington. Trawling, enhancing remineralization of sediment carbon. Yeah, that's it's a very interesting question. There is quite some discussion about. I have yes and no. There was a paper in Nature where they claimed, I have some problems with that paper because some of the assumptions they made are not. At least I'm not happy with them. But then there was no, not the budget. First, there are also observations that people did experimental work, and if you're doing a full meta-analysis, which some British colleagues did, basically you get no conclusive answer. Sometimes it is, sometimes it is not. That's the work that the people in Essex did. On Exeter in the UK. Also, when people did experimental resuspension in earlier days in terms of dredging, not so much fishing, there was also not a lot of strong evidence for this. But then if you're going into more longer timescales, if you're going to a system which from natural conditions, there's a lot of resuspension deposition. There you often see that the carbon burial efficiency, how much carbon survives, is far less than in steady accumulating sediments. So although the experimental evidence is not there, it may be that there is an effect of trawling, that it causes enhanced remineralization. But that's not, I haven't seen the evidence for it. It may be, but the evidence which has been reported, I'm not convinced about it. And there is a lot of wishful thinking for some people who would like to see it in order to argue for MPAs. And I don't think they did a good service for the scientific community, because you should be honest about it. And that's my answer, Ellen. I hope you're happy with it.
Yes, thank you, Jack. That's excellent.
Okay.
I see one more question from Fei Da. Do you want to unmute and ask your question?
Oh, yes. Yes, my question is, like you mentioned, PIC from rivers are important. Do we also need to consider like carbonate production in like seagrass meadows, all those in the budget analysis? Like I know there's some study in the Chesapeake Bay about that.
Yeah,
Thank you.
yeah, that's a good question. There's of course there is quite some carbonate production in the seagrass system. Anybody who has been snorkeling or scuba diving knows that some of the seagrasses have a nice whitish coverage, which is epiphytes, but also some calcium carbonate. But the picture I showed is really what is the river bringing to the marine, and it includes those coastal systems. So that's an internal cycling in the marine system. Also, I think the many mangrove systems are more sites of, say, carbonate dissolution than carbonate production. Tidal wetlands depends on where they are, and seagrasses as well. So it is really important to realize that both there is carbonate production in those systems, but there's also carbonate dissolution, and the balance is really important. And if you have a system which is intertidal, or where there's a lot of microphytes, which because of the oxygen loss by the roots and rhizomes produce strong acid in the sediments, you get a lot of carbonate dissolution, and that should be taken into account as well. But that's of course, we should look at it, and certainly regional, we should look at, if you make a detailed carbon budget for a coastal system, you should include not only the organic, but also the inorganic parts, and you should include local production as local consumption of inorganic carbonates. I agree with that. There is another one. Shall I, Marie? Okay, so the next question is from Marie Zeller. Is about the coupling between organic and inorganic carbon in the models. Yeah, that's something I didn't touch upon. That's a good question. There are many ways with the ballasting hypothesis, the shielding, and then if you are on the sediments, say respiration driven calcite, aerobic respiration driven calcite dissolution, or if you get organic matter mineralization on anaerobic conditions, you produce alkalinity and you can get carbonate precipitation. You cannot really uncouple them. But what I discussed today was mainly how we can develop very simple strategies to include some kind of benthic model to a pelagic model. And then you have to make choices, and you cannot include all those things into a simple model, because at the moment you start to really make a model where you have carbonate dissolution and precipitation in the sediment, you have to model your pH. And if you start to model the pH, then you, for those people who have done it, know how difficult it is to understand. And modeling pH in sediments, you have additional complexities that we do not know well how protons interact with the silicate minerals. And all the things that soil scientists have been doing for decades, we still have to learn that as marine scientists.
All right, well, thank you so much. We're at the end of our hour, and let's just all thank Jack Middleberg again for this great talk and discussion. We will be announcing our next webinar. We've tentatively scheduled for May, but announcement will be coming out shortly.
Okay.
Thank you, everybody, for joining.
Bye-bye.
Bye. Bye.